Hi kids, we're up to our next video, which is going to be on lipids. So this is our bio video number 4, our biochemical video number 4, and this is on lipids. All right, so lipids, I like to say they are still CHO, but lots of carbon and hydrogen and very little oxygen. Okay, so they still have carbon, hydrogen, oxygen, just like the carbohydrates did, but a lot more carbon and hydrogen and very little oxygen. Okay, lipids provide long-term stored energy. I like to say they're like putting a wet log on a fire, because the wet log is going to take a long time to burn, and it's going to need a lot of energy to make it burn. So like if you've ever had a fire in your backyard, I always say carbs are kind of like paper. That's like your sugars and stuff. They burn up fast, just like in your body, sugars do. Twigs are kind of like your simple sugars, like your disaccharides and all. And dry logs are kind of like your polysaccharides. They would burn, they would keep it going for a while. They provide you a lot of energy. They're the first thing that burns and stuff, and they last a little bit longer, so that's like pasta. This is why football players like to have pasta dinners before their events, their games, and why other sports also we want to get a lot of pasta in there, because we want to have those dry logs on our fires, okay? But lipids are like a wet log, okay? If you put a lot of fat into your body, it's going to take a lot to burn that. So you got to get, and if you put a wet log on a fire, it's going to take a lot to burn that log. So that's why we call them long-term stored energy, because they will burn, okay, but it will take a very long time. And usually, if you consider fats, and unfortunately some of us have to consider fats on our body, they're really tough to burn off. It's really tough to get off, okay? All right, the unifying feature of lipids is they're hydrophobic, okay? They have no affinity for water. They are not going to mix with water, okay? And these are going to be, you know, your fats, your oils, your triglycerides, phospholipids, and even the sterols. And you'll see what sterols are if you haven't heard of them before. Fats are constructed from two types of smaller molecules. We like to say these glycerol molecules, but really it's all about the fatty acids. Fats or lipids are not going to link up like our carbohydrates did, like where we had monosaccharides, right, linking up into polysaccharides, because fats start off and they're already big, kind of like this marker. It's already a big marker, so, you know, it's already a big molecule. So we're not going to see these molecules like linked together like the carbohydrates would, or even like the proteins will in the next video that we talk about. Instead, just really one fat is all you need. But will they link up? Will these fatty acids still be like a building block for something? Yes, they will, and we'll see that with triglycerides, phospholipids, and even waxes, okay? So think of our fats as like this, not just like these monomers, but they're big monomers, okay, and they're fatty acids, okay? And a fatty acid, we'll see the construction of a fatty acid or the composition of a fatty acid in, like, when we first see, like, a diagram of it, okay? All right, so that's, like, our beginning talk, but a little bit more, okay? A fatty acid, think fatty acid, right, has a carboxyl group at the end. That's where it gets the name acid. But the long hydrocarbon skeleton is the fatty part. So I always like to say this is the fatty part, and this is the acid, okay? Fatty acids, okay? They vary in the number of their carbons in that carbon skeleton. And we usually look at fatty acids as one of two types, saturated and unsaturated, okay? And here's where you might remember a lot of this from your first year bio. Saturated fatty acids. They're fatty acids because they're solid at room temperature. Another thing they're mostly animal, okay? They're a little tougher on the body to break down and get rid of, okay? And there's no double bonds in that whole hydrocarbon chain, okay? But unsaturated, on the other hand, so that was our saturated fatty acids, no double bonds. Unsaturated fatty acids, they're going to have one or more double bonds in that hydrocarbon chain. They're going to be liquid at room temperature versus solid at room temperature, and they're primarily from plants. Plants and some of your fishes are also your unsaturated ones. And they're typically called your oils. The unsaturated ones are oils, okay? And we'll get to the trans fats in a little bit. I don't know if it's the next slide or what. Okay, so going on to the next slide, if I can get this apart. We see a different kind of construction of our fatty acids here. If we look at this right here, this is butter. Oh, gotta love butter. Butter. Okay? Butter comes from an animal, right? So it's definitely going to have saturated fatty acids in it. This molecule right here is showing a triglyceride, but we're trying to focus on these fatty acids, these 3 fatty acids. This is the glycerol, 1 glycerol, 3 fatty acids. We'll see that again in another picture. But we're looking at these fatty acids here. Notice that there's no like kinkiness to them. They're very like stiff, kind of long, you know, structurally strong molecules, okay? Stearic acid is basically animal fat, okay? And that's like your saturated fats. But if you think of oils, what's the difference between saturated fats and unsaturated oils? The difference is in the fatty acids. There's a double bond in here, and what that does is it causes that fatty acid to kink, okay? And this double bond, we'll see about the cis and the trans in a moment, but this double bond causes it to kink and actually loosens up this. So when we look at oils, especially coming from plants, you know, the seeds of plants too, very fatty in the seeds, those oils, okay, are kind of kinky. That kinkiness, that double bond, you would think a double bond would be stronger, but it's not. It actually weakens that very strong, straight kind of look of a saturated thing. It kind of kinks it. That kink actually makes our bodies able to process it much more. So it won't like linger in our blood vessels and cause heart disease and all kinds of things like that. So there's another comparison between our saturated fats and our unsaturated oils or unsaturated fats. Okay? Now, what is the trans fat? We see this down here, and let's try to just look at this first. A diet rich in saturated fats may contribute to cardiovascular disease. Yeah. Hydrogenation is the process of converting unsaturated oils to saturated fats. And this is why we always used to say in first year bio, like hydrogenation. If you read on your ingredients and you see those words, partially hydrogenated oils. You'll see that a lot of times. Partially hydrogenated soybean oil, things like that. If you see that, you know, run, okay, because this is a trans fat, okay? They've been hydrogenated. We'll see what that means in a moment. Okay, hydrogenation obviously is going to transform, and that's what we used to say in first year bio. They're called trans fat because we transform an oil into a fat. Not true. It actually has to do with that cis and trans, if you remember, with geometric isomers. Okay, so let's see that. Here is an unsaturated fat, and by the way, this is a double bond here. I'm just going to draw it as a double bond like that, and it's kinky, right? Notice the hydrogens right around that double bond are both on the same side. That's cis, right? That's one of those geometric isomers where if they're on the same side, remember how I showed my thumbs, like if they're on the same side, that's cis, but if one's down or something like that, then that's trans. Here we have, there's the double bond again. I'll draw it like a little bit more prominent here. One's up, one's down, that's a trans. That's where the term trans fat actually comes from. And through that process of hydrogenation, makes it twist, makes that geometric isomer, okay? And here's the thing. When people first wanted to hydrogenate oils, they thought, oh, great, because when people would cook in oils, things would get like a little soppy with the oils, and it would look like kind of greasy, right? And people didn't like the look of that. And oils, because they're liquids at room temperature, they wouldn't stay on like French fries and stuff like that so much. Instead, the French fries would look soggy. So they thought, and they wanted to stay away from the saturated ones because they knew saturated ones, animal fats. And let me tell you, McDonald's used to like cook their French fries years ago in animal fats. Like, imagine like the grease that comes off of bacon after you cook it, you know how it turns white and it hardens. That's what they used to use to cook the French fries in. And it tasted good because the fats like solidified on there, it didn't make them look soggy, and that was more appealing of a taste and all. But we found out that that was causing a lot of cardiovascular disease. So they moved away from that, and they moved to the oils, but that was making things soggy. So what did they do? They transformed those oils, okay, to try and make them solidify at room temperature. And when you transformed it like this, it made it a little bit more stiff, okay? But because of that stiffness, it actually made it even tougher to break down in our bodies. So where we thought we were going to like cure heart disease by doing these trans fats, it ended up that heart disease like skyrocketed up, and it was like, okay, we got to stay away from your margarines. And this is like your oils, this is like your margarines, and butter is still like your good old time saturated fats. So stay away from the margarines, even the ones that say they're heart smart. Those margarines have a lot of trans fats in them, and that's what keeps them at like somewhat room temperature. Otherwise your margarine would be a puddle and all. And people try to take like so much of the trans fats out when they say, you know, oh, it doesn't have, or it has very little trans fats in it and everything. But then they put a lot more water in it and it goes back to being a soggy thing. Okay, so here's another picture of comparing the three types. Okay, and I like this picture a lot. Okay, you can see how with the saturated fats, very, very stiff, and all single bonds within those carbons, right? And here's that carboxyl, these are fatty acids. So here's the fatty part, here's the acid part. All fatty acids have this carboxyl group at the end that designates it as an acid. Here's unsaturated. Notice it is cis, excuse me, because two hydrogens are on the same side. That little kinkiness, and even this kinkiness, this is a polyunsaturated fat then, because just having one more than one, okay, just having one makes it unsaturated, having more than one makes it polyunsaturated. But anyway, that makes it extra kinky. Excuse me again. Okay, now trans, what did they do? They switched these hydrogens. Notice it's stiffened up, and you might be thinking, well, this is more stiff than this. Yes, it does look that way, but because of this and because of the straightness of this double bond actually has a little more oomph to it, so it is a little tougher to break down. So you got to stay away from those trans fats. They really can clog up your arteries. Okay, so next slide. So now, how do we polymerize our lipids? Remember I said they're so big, they're this big. We're not going to like link them up in chains like we would like the monosaccharides. It's just going to make it too big and too unsteady. So what do? We usually link these lipids or these fatty acids to like a backbone. Okay, so imagine like three of these hooked to like some sort of backbone. Okay, we'll put my whiteout pen here. They're hooked to some sort of backbone. We have three of these hooked to a backbone. This backbone is that glycerol molecule. And we'll see with the triglyceride, there's three fatty acids to one glycerol molecule. With the phospholipids, we'll see that there's two fatty acids, with all my office supplies here, a phosphate group and that glycerol. And then we'll see with waxes, you got something totally different. You got this long alcohol attached to one fatty acid, okay? And then sterols are a completely different case altogether. So let's look at the triglycerides first. Three fatty acids to one glycerol, a 3-to-1 ratio. Phospholipids, two fatty acids, okay, one phosphate group and one glycerol, a 2-to-1-to-1. I always say 2-to-1 still with an additional phosphate group. There's one of those functional groups again. And waxes, you got one fatty acid, one, attached to one long alcohol, okay? This makes them highly waterproof. This is why waxes are used for like lip balms and car waxes and wax, like if anybody wants going to be a plumber, got these big wax rings that they put around before they install toilets and stuff. And then steriles are in another group altogether. We'll look at that in a moment. Okay, let's look at some of these molecules. Okay, now here is a formation of a triglyceride, and guess how we're going to link things together? Well, just like in the carbohydrates, when we linked monosaccharides together, right, it was by removing water, and it was called dehydration synthesis. Well, each fatty acid is going to get linked onto this glycerol molecule. Here's the glycerol molecule by dehydration synthesis. The OH of that carboxyl group and one of the OHs from the glycerol, or the H from that OH, remove water, and then what we call an ester linkage. An ester linkage, that's the bond between fatty acids and the glycerol, that ester linkage right there. See how it goes, that CO, the O came from here and hooked on, okay? That's an ester linkage because of the dehydration, or the, dehydration synthesis, okay? But we're going to hook 3 onto there. See, 1, 2, 3. Quick question. Take a look at these fatty acids. Are they saturated? Are they unsaturated? Are they trans? Moment here to think. And you're right. They're saturated because there's no double bonds in those hydrocarbon chains. Okay, one double bond would cause kinkage, and that would be unsaturated. And whether it was cis or trans would depend on where those hydrogens are. Okay, but I want to show you that is a triglyceride. So on this is a triglyceride. Okay, and that's the first type of polymer when it comes to lipids. Okay, here's just another picture of that, but notice this one here. These two are saturated, this one is unsaturated. Is this cis or trans? Moment to think. And since the hydrogens are both on the same side, cis. Okay, this is truly a triglyceride that would come from a plant most likely, or a fish. Okay, it has that kinkiness, it can break down better, it's better for you, it's probably a liquid at room temperature. It's just that one little double bond there, causes all that to change so much. All right, now the next type of polymer form. It's the phospholipid. Again, in this diagram here is the glycerol molecule in here. And we have 1, 2 fatty acids attached, but in the third position, instead of having another fatty acid, we attach this phosphate group, okay? And it has this little head, this choline head. Don't worry about that so much. If you know that it has a phosphate group and 2 fatty acids, I'm good with that, attached to a glycerol, okay? Now, let's take a look at these fatty acids. What are you thinking about them? Yep. One is saturated, one is unsaturated. So as long as there's one unsaturated in there, this is considered unsaturated. As long as there's one thing, either in this or in this, same with that triglyceride back there. As long as there's one little kinky part in it, then it's considered unsaturated. Now, because you can't quite tell if the hydrogens are cis, like on the same side, or trans, I wouldn't ask you that so much, but I will tell you, because it is kinked, most likely this is cis. So 2 of the hydrogens are coming off on the same side here. Okay, it would be a little straighter, then it would most likely be trans. Okay, and when we draw phospholipids later, when we get into like cell membranes, we always draw them with this head. Here is that head that's hydrophilic. There's that word, hydrophilic, loves water. Okay, with the phosphate and the glycerol and this little choline thing, and then the 2 hydrophobic tails. Now, because these tails actually are so much more a part of this molecule, that's why fats in general, phospholipids, are very hydrophobic in general. Like, even though the head is hydrophilic, most of this molecule is hydrophobic. Okay? And finally, our waxes and our steriles. Waxes, you have, here is kind of representing the wax structure. This is like the long alcohol end, so this is like the alcohol part of it, alcohol. This is not a great picture. here is the fatty acid part. And you can see with waxes, it's one fatty acid attached to an alcohol group. That's about all, except for like that they're waterproof, that I need you to know for that. Now, sterile structure, sterile, okay, has this 5-sided thing, 6-sided thing, 6, 6, 6, 6, 6, 5, okay? Then the rest of it depends on what kind of sterile you have. This sterile here, I'm not quite sure what that sterile is there, okay? But look at the next page. We look at 2 steriles, and they happen to be hormones. A lot of your hormones that are lipid-based, that are sterile-based, like your testosterone and your estrogen, if they're ending in "-one" or "n", like "en", not "ine", or not "in", simply "in", they're sterile-based. Look at the difference. Testosterone is the male hormone. Estradiol, okay, which is estrogen basically, is the female hormone. Look at the difference, okay? Not much. Testosterone has an additional has an additional methyl group down here, right here. Estrogen's missing that. And testosterone has a little carbonyl group here versus estrogen has an alcohol group or a hydroxyl group here. Now you might be saying like, oh, there's an HHH here, HHH, and there's one up here. There's actually one up here, if you see this black with the red showing OH, but it's kind of coming forward, so it's hard to see that on there. But that is another H. So really the only differences between testosterone and estrogen are the hydroxide for a carbonyl and the appearance of that methyl group or, no methyl group. All right. So, but you can see they have that basic 6665 structure of like that all sterols have. And sterols are not just some of your hormones, but they're also your anabolic steroids are sterile based. So is cholesterol. That's why it has sterol in it. Okay. And we'll see what cholesterol plays. A lot of your sterols are involved in like information transfer in the body and identification, so we'll see later with that. So that's it for our lipids right now, and that's it for this video, okay?